Hi, everyone. Good evening. Uh, a warm welcome to all of you here in person and those who join us on Zoom. Welcome to our first in-person Science Cafe. My name is Ye Duan, I also go by Oscar. I'm a postdoc researcher at the Harvard University and also UMass Chan Medical School here in Worcester. I serve as the co-chair of the outreach committee of UMass Chan Postdoc Association, along with my colleague Alessia and Ruchi. We're gonna see in a minute. Uh, and we started our Science Cafe back in 2020 via Zoom with the goal of bringing the excitement of biomedical research taking place at UMass Chan to the community. It's about making science accessible, sharing our discoveries, and engaging in discussion with the public, which is you all, and also encouraging curiosity in the community. In the past few years, we've hosted 20 sessions out on Zoom, and you can watch all our past Science Cafe by searching UMass Chan Postdoc Association on YouTube or by scanning the QR code on the slide. So you have 10 seconds. 5, 4, 3, 2, 1. Okay. And tonight marks a milestone as we are hosting our first ever hybrid Science Cafe with both in person and virtually on Zoom. And this is also our first collaboration with the outreach program of Department of Biochemistry and Molecular Biotechnology of UMass Medical School. So before we begin, I'd like to extend a thank you to Dr. Sean Ryder and Dr. Haley Barlow for their tremendous help, especially Sean for his invaluable tech support. We also like to thank WCUW for generously providing this wonderful space for our event. And we would like to thank the OMPA co-president, Mina, for her help for organizing this event. So before we start introducing our speakers, I would like to invite Sean on the stage to have a few sentences.
So for those of you who don't know me, either in person or online, my name is Sean Ryder. I'm a professor and the vice chair for outreach in the Department of Biochemistry and Molecular Biotechnology at UMass Chan Medical School. I got involved in outreach efforts through my department because I feel like as a scientist, it's our privilege and duty to be able to perform experiments and to learn new things. And it's our duty to be part of the community, to listen to our I don't want to say constituents, but neighbors, the people who support us, the people who surround us. And I'm really interested in building bridges between the science that happens on campus and the people that live in the community that surrounds us. And I'd very much like to echo Oscar and thank WCUW for allowing us to use this beautifully renovated front room area for this first hybrid in-person online science cafe. I'm so grateful to be collaborating with the UMass Postdoc Association. They do all the hard work. I just twiddle knobs back on the soundboard. Makes my life super easy. And thank you all for coming. And I'll turn it back over to Oscar.
Thank you, Sean. Also, before we introduce our speakers, please note that the opinions expressed during the presentations are those of the speakers and do not necessarily reflect the views of UMass Chan Medical School. And also, we have a quick reminder about the Q&A session. So if you have questions and you're attending in person, please wait for the end of each talk, and after the talk, please raise your hand. We're gonna deliver the microphone to you. And for those who join on Zoom, you are welcome to type in your questions in the chat box, and we will read it out on behalf of you at the end of the talk and in the Q&A session. And also, we kindly ask everyone to use clear and accessible language and avoid using technical jargons or abbreviations so that everyone can follow and enjoy the conversation. And finally, this session is being recorded and will be uploaded to the YouTube channel. So feel free to share it with your friends or families. And also make sure to follow our social media channels, which are listed on the screen and in the Zoom chat box. So let's get started. Our first speaker is Dr. Sneha Shah, and Dr. Shah did her PhD in Oxford University in UK, followed a short postdoc at TIFR in India and Harvard Medical School. And before and after that, she joined Joe Richter's lab at the UMass Chan Medical School here in Worcester. Her scientific journey has focused on studying how DNA influences cell function using simple organisms like yeast. And then she moved to mice and now human cells and tissues. Apart from the lab, Sneha loves spending quality time with her toddler, which is in the audience, very cute. Getting him to love going on hikes, building things, doing crafts, and so on. And as a family, they love traveling and exploring trails in New England. So Sneha, the floor is yours.
Hello, everybody, and thank you, Oscar, for that kind introduction. I am very excited to present to you the work that we've been doing. Let me get my slides up here. Is that good? All right. So I'm Sneha. I'm an assistant professor in Joel Richter's lab at UMass Chan Medical School. And I'm excited to share with you our work on designing an RNA therapy for Fragile X syndrome. So Fragile X syndrome is a neurodevelopmental disorder. Children around the age of 2 begin to show symptoms of Fragile X syndrome, which include delays in speech and motor skills. Some of these patients with Fragile X are nonverbal throughout their lifespan. The symptoms then go on to show behavior. Patients then go on to show behavioral issues like hyperactivity, anxiety, and some of them also develop seizures. This syndrome lies on the autism spectrum disorder. 50% of Fragile X patients do develop autism as well. And currently, there is no FDA-approved treatment. So patients with Fragile X, those with very severe manifestation of this disorder, require lifetime caregiver assistance and support. And these, the treatments that are currently in clinical trials are those for symptomatic relief. So what is Fragile X syndrome? When scientists began to study this in the late 1900s, they found that this disorder is passed on from the parents to the child, which meant that the gene, the problem that is present is on the chromosomes or the DNA of the parent that is passed on to the child. And what I'm showing you here is the X chromosome, the X chromosome here. So X chromosome is one of the chromosomes that is present in our cells. In males, there is only one X chromosome present, but in females, there are two X chromosomes. Each chromosome is a collection of genetic information in our cells. And what they saw was that in the X chromosomes of Fragile X individuals, there was a site over here which looked like it was just going to snip off. So that is how the name Fragile X syndrome also comes around. And because there's only one X chromosome in males, if a mutated version of this X chromosome is present, the males tend to show more severe phenotypes, and it also occurs more commonly in males. In females, because there are two copies of the X chromosome, if one is not mutated, you would not see symptoms of Fragile X manifest as much. So to understand what's going on at this region of the X chromosome, let me introduce to you the central dogma of molecular biology. What that means is the chromosome that I just showed you has small packets of information lined up on it, which is called genes. Each gene has all the information that is required to make proteins. This information in a gene is read in the form of an RNA. So that's the information that's required to make the final product, which is a protein. To give you an analogy for this, you can think of the gene or the DNA as a cookbook. It has multiple recipes lined up in it. And if you wanted to make a cupcake, you would read the recipe for it, make the cupcake, and a collection of different products that are made from every recipe or every gene results in proteins that function, that come together to make the cell function properly. So to take this a step further, what can also happen is you want a different flavor of a cupcake. So for the same gene, you can actually have multiple recipes that have certain small tweaks and changes that can result in many different proteins. So here I'm introducing to you the concept that one gene can have multiple RNAs. So multiple ways in which the same gene is being read to make multiple different proteins. This is called alternative splicing of RNAs. Now let's go back to Fragile X syndrome. When scientists saw this region, which looks fragile on the X chromosome, they wanted to find out what is the packet of DNA that is present over here, what is the gene that is present. And in 1991, an international group of scientists found that there is a particular gene that is mutated or incorrect in this region of the X chromosome. They called it the FMR1 gene. This gene, or this cookbook, is read in the form of the FMR1 RNA, which is the recipe, and it makes the protein, which is FMRP. So the function of the FMR1 gene is to make the FMR1 RNA and the FMRP protein. FMRP protein is a very important protein present in all our cells and does many different things. So if the FMRP protein is not present, it does lead to many problems, which results in Fragile X syndrome. So what goes wrong in the FMR1 gene in a Fragile X individual? So let's look at the gene more in detail. So this is the FMR1 gene. And what it contains is, at the beginning of the gene, these regions which are called repeats. So the same sequence of RNA is repeated 55 times in normal individuals. So it can range between 6 to 55 times. And when this range of repeats is present, we get normal levels of FMR1 RNA, normal levels of FMRP. But in Fragile X individuals, what they found was these repeats are replicated greater than 200 times. So the repeats are expanded. This results in loss of FMR1 RNA in most patients and loss of the FMRP protein. And also because of the gene mutation, you start seeing this appearance of the chromosome, which looks very fragile. So this is interesting because now we have a single gene that we can work on to address Fragile X syndrome. So what we wanted to ask was, what happens when the FMR1 gene is mutated in patients? And to answer this question, we collaborated with Dr. Liz Berry-Kravis. She's at Rush University Medical Center and runs a fantastic Fragile X clinic. She sent us blood samples from 29 Fragile X males and 13 typically developing or control age-matched males. We got these blood samples. We extracted RNA, which was the recipe in the analogy that we spoke about earlier. We purified this RNA, so we now have only RNA and nothing else present from the cells. And then we studied the sequence of this RNA. What does this RNA look like between Fragile X individuals and control or TD samples? And I'm going to show you a trace of the data now. So what we're looking at here is the FMR1 gene. And to remind you, the FMR1 gene is mutated in Fragile X individuals. It has a lot of repeats. And in most individuals, it is believed that there was no FMR1 RNA and no FMRP protein. But there are instances known where some individuals may still express some RNA, and we'll get to that. What you're looking at here is a cartoon of this FMR1 gene that I've shown you here. And the way to read this is the vertical lines in this entire FMR1 gene are called exons. This is a packet of information that is important in the recipe to make the correct protein. The remaining horizontal lines are called introns. This is the part of the gene that is not required and is usually cut and removed out or omitted. Below here in blue are the FMR1 RNA reads in the control or TD. So every spike is essentially the portion of the FMR1 RNA that is being made in the control samples. So that's what you're seeing in blue. They line up very nicely with the exons, which are the regions I told you are required to make the correct FMR1 RNA. Next, I'm going to show you traces of 2 Fragile X individuals. So those are the ones you see now. The first is from a Fragile X1 person, and the second is from a Fragile X2 person. And here I would like you to get involved and help me spot the differences between the FMR1 RNA in these red Fragile X individuals and the blue control individuals. So anyone take me up on this challenge? One difference, two differences, whatever you would like to point out. You do.
It's right there.
Yes, that's good.
The big one.
That is a big one. And another one? So we've got two Fragile X and one control. Yes, please.
Looks like someone forgot to put the data in number one.
Exactly. Thank you so much. So the differences that were pointed out by our fantastic audience is there are two groups of Fragile X individuals that we're looking at here. The first one is represented by this FXS1 individual who has no FMR1 RNA. Now, this would be similar to what I spoke to you earlier about, that in a mutant FMR1 gene, there is no RNA and no protein. So that fits with what was known. But the second individual is actually making a lot of FMR1 RNA. There were only few patients that were known earlier to make this FMR1 RNA, and that's what we're seeing here. So there are two groups of Fragile X individuals, but what was very interesting is this. So there is a region here which is made in only this Fragile X individual that makes the FMR1 RNA, but not in the control individual. So now let's take this a step further. And now I'm showing you all the data we had from all the patients that we look at to just see the magnitude of this data that I'm talking to you about. So the first set of 13 individuals here are the controls. You can see really nice exon definition, that is only the exons are being made. But in all the Fragile X individuals now, there are these two sets of patients that I spoke to you about. The first one is the 70% of them are making the FMR1 RNA. So you see these exons being made. And the second set is this group down below here, where there's no FMR1 RNA. So now we are saying that unlike what was previously thought that most individuals do not make RNA, in the set of samples that we've looked at, most individuals are actually making, most Fragile X individuals are making the RNA. And there's only a small subset that is not making the FMR1 RNA. But what is very interesting is the fact that in the Fragile X individual samples that we looked at, there is transcript being made in this region here in the intron. This is very specific. It's not there in any of the controls, only in the Fragile X individuals, and only in the ones that make some amount of RNA. So what I'm showing you now is in our data set, we find typically developing or control individuals make the full FMR1 RNA, and we know that they make FMRP protein. We've tested that too. But in Fragile X individuals that we've looked at, there are two types of RNA that come up at least. One of them is the full-length RNA, the FMR1 RNA. But the second one, and the most prominent, so 60 to 90% of the RNA that they make is this form which is shorter, the incorrect form that's made from the intronic region that ideally should have cut and been omitted, but it is included in these patients. So these patients are now making this small transcript, which has, which ends right here and cannot make the full-length FMR1 RNA, therefore does not have all the information in the recipe to make the cupcake correctly. So we cannot get full-length FMRP protein. So therefore, even though the Fragile X individuals are making some RNA, because most of it is wrong, we are not able to get enough FMRP protein, and they have Fragile X symptoms. So this is very exciting because now we found something that is specific to a Fragile X individual, and maybe we can use it to correct things. And that's where I'm going next. So the next thing we asked was, can we switch the expression? Can we switch from that incorrect form that does not have all the information to the correct form in the FMR1 Fragile X individuals? So to do this, I want to introduce you to a class of molecules that is being used for this kind of correction. These are called antisense oligonucleotides. So these are programmable drugs, very small 20 nucleotide oligos. So they can be either DNA or RNA that are chemically synthesized and can be modified to produce different effects. So ASOs are very specific to your gene of interest. So they can be extremely targeted. And you can have them for different functions in the row in the cells based on where they are targeting and what their chemistry is. So for example, you can have ASOs that go and bind to your target RNA. So remember, these are 20, 21 nucleotide small oligos, but your RNA of interest is around, on average, 1,600 to 2,000 nucleotides. So it's a very small piece of RNA or DNA that can be very specifically targeted. In these cases where it can go and target this RNA, and based on the chemistry, it can recruit certain enzymes or proteins that chop up that RNA. So you can actually degrade your target RNA if you use this chemistry. Another kind of ASO could be the one that goes and binds to certain regions of the RNA and stops machinery that recognizes that RNA to make protein, because I said the RNA is read in a way that it can make protein, but you can block that by putting an ASO there, and the RNA is no longer read to make protein. But the third class of category is also the one which can switch splicing. So here what you can do is target your ASO to a region which you do not want to be read. So now it does not read that region. It goes to the next packet of information on that gene and reads that instead. So you can switch which RNA is being made. Now, there are a few FDA-approved ASOs in the clinic now for many different disorders. And for example, for SMA, the Spinraza is an ASO that is very effective, and therefore there's a precedent of using it for different neurological disorders. The ASOs that are in the clinic have low toxicity, and one of the main benefits is it has good brain distribution. So I told you Fragile X is a neurodevelopmental disorder, so we do want to help increase the FMRP or levels in the brain. And with this ASO, with ASOs, you can put them in your cerebrospinal fluids, CSF, which bathes the entire brain. So if you put the ASO in, it may go and be delivered into different cells of the brain. So that is a possibility. And then if it is safe and effective, you could repeatedly dose individuals based on what treatment regimen is required. So amongst these, the ones that I would be interested in is the splice switching, because what I want to do is change the expression of this incorrect RNA and switch it to the correct RNA. So here we are going to use ASOs to do that. And ASOs for this project were designed and synthesized by our genius ASO expert in UMass Chan Medical School at the RTI, Dr. Jonathan Watts. He made these fantastic ASOs that we've tested in our cells. And I'm going to show you a snippet of the data. So to recap, we have Fragile X individuals that have a mutant FMR1 gene. They're making high amounts of this incorrect transcript, the shorter one, and low amounts of this FMR1 RNA, due to which they cannot make enough FMRP protein. We took these Fragile X cells and added the ASOs to see if we can change the expression of the RNA. And what you're looking at here on the left is Fragile X cells. Each dot represents one replicate. So there are many cells that we, or many samples that we've tested. The first set is without the ASO, and the second is with the ASO. And what we wanted to see is more of the full-length RNA, less of the short RNA. And that's exactly what we saw. The full-length RNA levels increased. So you see more of the full-length RNA being made after ASO treatment. And the stars here, or the asterisks, signify significance. So we've put this through multiple statistical testing, and this is highly significant. So we are getting significant increase in the amount of correct FMR1 RNA. But at the same time, we are also getting a decrease in the incorrect RNA. And this is exactly what we wanted to do, switch the splicing. So now we're getting more of the full-length RNA, less of the short RNA. And if the FMR1 RNA is recovered, do we recover the FMRP protein? So what you're looking at now is Western blot data or protein expression data. Essentially, the band on this blot here represents FMRP levels. And if you look at without ASO and plus ASO, you see a clear increase in FMRP levels. So what we are showing is with ASO treatment in Fragile X cells, we are able to decrease the incorrect RNA, increase the correct RNA, and restore FMRP protein levels. So this becomes really exciting because this could be a therapeutic target. There's a lot of work still to be done, but essentially what we would hope with this approach is in the future, an ASO targeting this incorrect FMR1 RNA could be used to restore FMRP levels in patients. But this is just the beginning. There's a long road ahead. There's a lot of work that needs to be done because we need to understand how the ASOs act. What is the function of that small transcript that is being made or the small RNA that is being made in Fragile X individuals? Which is the best ASO to use? Because we still have to test which, if they are toxic, if they are good to use, if what is the treatment duration, and so on. We also need to know, so this was done in cell lines, which are blood cells, but I told you this is a neurodevelopmental disorder. So we do need to test in the neurons how this works. So this is a pilot experiment just to say that this approach may be beneficial, but we have a lot of work to do before it can be taken to the clinic. And one of the most important things for Fragile X becomes, what are the markers? How do you know that the ASO that you put in is functional and is leading to any change in the patients? So another part of my project is studying biomarkers for Fragile X so that we can test how it's functioning. This work that I presented today was published in a PNAS article a couple of years back. And with that, I'd like to thank everybody that's been involved in this project. It's been a fantastic group effort. But first and foremost, I'd like to thank the patients and their caregivers who gave us this, the precious samples for research, which is very important for the work that we do. And most of this work was done as a fantastic collaboration between our lab. So this is Joel, my guide and mentor, and he's been a fantastic support system through this entire project. Liz Berry-Kravis, whom we got the Fragile X patient samples from. She's an excellent clinician. And John Watts, who's our ASO guru, whom we work with. And then this is our Richter lab. And without the support of a fantastic lab, you cannot do any of the work that you do. It does take a village to actually do science. And Maria, Suna, Mina, Dan, they've been fantastic through this entire project. And we do have funding from a lot of sources. One of the most important things to conduct science and the work that we do. So I do thank all the funding sources that have been instrumental for this project. And for today's session, I'd like to thank UMPA, BMB, and of course, WCUB for hosting, CUW for hosting us here. And all the audience present in person and on Zoom, thank you so much for taking your time out to listen to me. Thank you. Happy to take any questions.
Yeah, that's a question.
Sure.
Do you have any idea? Hold on a second. Oh,
This microphone for you.
Was on Zoom. Yeah, thanks for it. Was really lovely to see the talk and the work you presented. I was curious if you have an understanding of or an idea or hypothesis about why some of the patients had no RNA and others had this messed up RNA. I wonder if is there any understanding of that?
Yeah, thank you for that question. That's an excellent one. So this actually brings us back to how this FMR1 mutation works. So what we know is when you have this expanded repeat, there is something called DNA methylation that occurs on the entire gene. So DNA methylation is certain modifications of that DNA which shut it off. Now, no RNA can be made from it. And therefore, you do not get FMR1 RNA in most individuals. But in some individuals, what we think happens is there is less of that methylation. So that mark that stops that DNA from being read is reduced. And because of that, that RNA can still be made from this full mutated individual. And we did check methylation, the DNA methylation, the marks that stop that DNA from being read. And we do find that in individuals that make the RNA, there is less of that mark. So there is some other signaling that is going on at the same time that determines whether that gene is going to be read or not. Thank you.
Hi, great presentation. I do have a quick question about ASOs that you're using. You said the length is about 20 to 21 units long. Is it possible that that ASO could also react with other locations in the DNA?
That's a fantastic question. So the way these ASOs are synthesized is usually they run through a program which checks all the sequences that are present in our genome. So it will try to match it up everywhere. And we do end up using ASOs that have low capacity to match other regions. But that is not to say that it will not match at all, because it may not be 100% match, but maybe a 99% match, and that will still target another region of the DNA. So one of the things that we do need to do is, after ASO treatment, check all the genes. So we are going to do RNA sequencing and check whether other RNAs are being affected too. So those are important things that we do need to assess. Thank you.
I also have a question about the antisense oligo. So after one ASO hybrid, like
Base pairing.
base pairing with the target and triggers the degradation of the target RNA, what happened to that ASO? Will it target another molecule of the RNA, or it just gonna die with the target?
Thank you. So that is actually two different things, because the ASOs that we use, we think they are only switching splicing. So they are not the ones that can degrade, that can recruit things that can degrade the RNA. So we think the RNA is still intact. It's just that the splicing is happening at a different position. But on your note of the other ASOs that do bring in machinery that degrades, the ASOs are such that the middle of the ASO, it's called gapmer ASOs, where the middle of the ASO is a DNA and the sides are an RNA. So that RNA stops the ASO itself from being degraded. But because there's a DNA RNA hybrid in the middle, the machinery that degrades that RNA is being recruited. So the ASO itself is protected from degradation and does hang around in the cell for longer. And that's what you need for a drug as well, that it should be more stable. So the ASOs themselves are stable, but the target RNA in that case would be degraded.
Yeah. Hi, Sneha. It's beautiful.
Sorry.
Go ahead. Yeah, I just want to ask that it seems that it is curable. So what do you suggest that what will be the right age for the patient to get?
Fantastic question again. So this is still, of course, a pilot experiment in cells, so very far away from patients right now. But the idea would be the earlier, the better. But of course, that's very difficult because we do have to then test in children, and that's difficult to do. One of the problems that we do face is this particular syndrome. We do not have a mouse model for this. So we do have FMR1 knockout mouse models where the gene is just not made. But in mice, you don't have the sequence that makes this incorrect RNA. So I do not have a replicate in mice where I could have tested different ages and so on. So unfortunately, I don't have that, but we do in vitro neurons. So we take patient cells and convert them into making neurons. And we do believe that the earlier you target and the earlier you remove the RNA, it's going to be better. But we do not know that yet.
What is the age of manifestation of symptoms in the patient?
So around the age of 2, because that's when speech develops anyway. So you can see the delay. Therefore, around the age of 2, it's tested.
And you mentioned like 2 variants of FMR1. So is there like phenotypic difference between the patient? Did you observe or any
a little
comments?
bit? So we do see some mild correlation with IQ, but this is very small as a subset of patients. It's only 29. We need better statistical power to really say anything more than that.
Thank you.
Thank you.
Any other questions?
Yeah, we didn't receive any question from the Zoom audience. If you have any question from the Zoom, you can type in the chat box and we can read out for you.
All right. Thank you, Sneha.
Thank you very much.
You want to answer? There's a question in the chat, and a question. Could this be used to treat patients with FTD?
So to go.
That's a great question, but that is going to the next speaker. So Sandra's going to talk about FTD, and I'll let her take it. Thank you.
Thank you, Sneha. It was a beautiful talk. So with that, I would like to invite our second speaker, which is Dr. Sandra Almeida. Dr. Almeida did her undergraduate and graduate study at the University of Coimbra, Portugal. Upon receiving her PhD in cellular biology, she moved to San Francisco to conduct her postdoctoral studies at the Goldstone Institute, University of California. And there she was interested in studying frontotemporal dementia, that is FTD. She'll talk about that disease in detail today. She also learned how to perform cellular reprogramming from the member of the Nobel laureate's lab, Dr. Shinya Yamanaka's laboratory. Sandra moved to UMass Chen Medical School about 15 years ago, and she is currently working as a research professor and studying the FTD and other neurological disorders. When she is not in the laboratory, she enjoys to go walk with the family and her husband and 2 kids. The family likes to catch and release the frogs and other kinds of bugs as well. She also enjoys baking challenging birthday cakes. And her most recent culinary accomplishment is the platypus with Harry Potter glasses and scarf for her daughter's birthday. That's really interesting. If Sandra had not become a scientist, she would likely have become a baker. With that, I would like to invite Dr. Sandra.
Thank you. Thank you very much. Just gonna set up my presentation. All right. Hello, everyone. Thank you so much for the kind introduction. And I'm really excited to be here today to tell you some about the work that we do in the lab. Today, I'm going to be talking to you about the neurological disorder called FTD and ALS, which is kind of a neurological disorder it has 2 components. So FTD stands for frontotemporal dementia, which is the most common cause of dementia under the age of 60. This is really important because most people that have dementia, they are older than 65. So this is an early onset type of dementia. Most people that have FTD, they have ages between 45 and 64, which is really very young people. This is a progressive neurodegenerative disease, which means that once it starts, it continues to get worse. After diagnosis, people typically have 7 to 13 years left to live. And unfortunately, there's no cure or treatments available for this disease. So as you can probably guess from the name of the disease is caused by loss of brain cells in the frontal and temporal lobe. And these regions are important for a lot of functions. And so when cells die in these regions of the brain shrinks, and that's when the symptoms start. So the symptoms that people develop depend on the region, the extent the brain is affected. But the symptoms can go into 2 main groups. So they could be behavioral changes or language issues. Some of examples of behavioral changes are inappropriate social behavior. These people, they don't care about the others, poor judgment, and changes in personality. If the language is affected, so these people can lose their ability to speak, read, write, and even to understand what other people are saying. In addition to these 2 groups of changes, people can also have alterations in the movement. The other disease that I would like to tell you about is called amyotrophic lateral sclerosis, or ALS. This is another progressive neurodegenerative disease. Again, once it starts, it continues to get worse. And this disease is caused by loss of cells into regions, the motor cortex and spinal cord, which are regions important for movement. So when cells die in these regions of the brain loses control over the muscles, And so the people can lose their ability to walk, to speak, to eat, and even to breathe. Most people that have ALS, they are between the ages of 50 and 70, but can also develop the disease in the 20s and 30s, so very early on. And this disease progresses really quickly. So most people after diagnosis, they have 2 to 5 years left to live. So FTD and ALS kind of sounds like two completely different diseases, but we now see them as kind of a spectrum. So FTD and ALS are the two ends of the spectrum of disease. And if people lose cells in regions that normally are affected in FTD, as well in regions that are affected in ALS, so it's a mix, a combination, those people are diagnosed with FTD-ALS. For the most part, we don't know why those cells die in the brain. There's a small percentage of the cases where we think there's a genetic cause. And the most common genetic cause for both diseases is a mutation or an alteration in this gene called C9 or 72. So for the remaining of the talk, I will be just focusing on FTD-ALS caused by C9 or 72. This is kind of probably familiar to you based on the previous presentation. So most people have 2 to 3 units, repeat units, in the gene. They could go up to 20. But people that have FTD-ALS, they typically have up to thousands of these repeat units. And so I want to tell you what we know, how these repeat units can cause the disease. But before that, I want to tell you about how we study the disease. So this is a neurological disease, and ideally we would use brain, but people are still alive, so can't use their brain. But we have a way to use some of their skin, convert them into brain cells, and use them in the lab to study the disease. And I want to show you or tell you how that works. So everything starts with a skin biopsy, which can be collected in the doctor's office. We take the skin into the lab. We put a small piece of skin in a Petri dish, and we just grow the skin, the pointer. We can collect the skins that grow out of the little piece of skin. We then use reprogramming factors to make them into stem cells. These are very special type of stem cells because these stem cells, they have the ability to differentiate in any type of cell of the body. Because we study neurological disease, we use differentiated molecules to make them into brain cells. Here you can see that a type of brain cells are neurons, and so this is a picture of some of the neurons that we obtain all the way back from a little piece of skin. And so now we have these neurons in the Petri dish, and we can use them to study the disease. Understand, in this case, if you have a little skin from a person that has the C9 mutation, now we can study how this mutation can cause a disease. And so how exactly, this mutation called disease? Before that, I'm going to tell a little bit of what we know about what the gene does. So this is a simple representation of a cell, and a cell has two main compartments. It has the nucleus and it has the cytoplasm. The nucleus is where our DNA is, and the DNA codes or has information to make a human being. The DNA is made of thousands of genes. For instance, the C9 or 72 is one of those genes. And the genes contain the information to make a protein. So C9 gene makes C9 protein. But the factories that make protein are in the cytoplasm, and information to make a protein is in the nucleus. And the genes and the DNA, they never leave the nucleus. So we need a third molecule, which is called RNA, which then goes from the nucleus to the cytoplasm and can be used as the instructions to make the protein. The C9 protein works in a recycling center. So it's one of many proteins that work in a recycling center. And the role of this recycling center is to collect waste that is produced from the cell, and then it's going to be recycled and the cell gets rid of it. So C9 works in this recycling center. That's the function of the gene. In the disease situation, as you remember, we have these repeat units. And the presence of these repeat units is going to cause 3 consequences that I will be telling you about in a second. So one of them is because you have these repeat units over here, it becomes more difficult for the cell to make copies of RNA. So it means they're going to have less RNA. Less RNA means less protein. And if you have less protein, we believe that this recycling center becomes less effective. So over time, there'll be accumulation of waste in the cell, which is not good for the cell. So one of the consequences is that there'll be less C9 protein function, which could lead to accumulation of waste in the cell. The second consequence is that the cell can also make the RNA copies that contain the repeat units. And for a reason that we don't understand, they tend to accumulate in the cell, mostly in the nucleus, but also in the cytoplasm. When they accumulate in the cell, we think they're going to interfere with the normal function of the cell, so they become toxic. So a second consequence would be accumulation of these disease RNAs. And I would like to show you how they look like in cells. So here is a representation of a neuron, so one of our many brain cells. In green, you can see it's the cytoplasm, and in blue is the nucleus. This is a control human neuron that we differentiated all the way back from a small teen cell. And this is the neurons from a person that has the C9 mutation. And so what we did was we label those disease RNAs, and you can see them here in red. So these red dots are accumulation of this disease RNA. And I mentioned to you, so they mostly accumulate in the nucleus, but can also be in the cytoplasm. So I want to tell you about the third consequence is that some of these RNAs can go to the protein factory, and this region contains the repeats can be made into protein. These are abnormal proteins. We don't know what they do, but they tend to accumulate in the cell, and from many studies we have done, they appear to be toxic. And so a third consequence will be accumulation of these disease proteins. And so I want to show you how they look like. So this is an autopsy brain tissue from a person that had a C9 disease. And we have a way to label these proteins. And so as you can see here, there's accumulation of these proteins outside the cell. So we can actually, find these cells in the brain of people. So I just told you that these mutations can lead to 3 consequences. The first one, it's kind of hard to fix, so I will not talk about these today. But the other 2, the accumulation of disease RNA and accumulation of disease proteins, which we think are probably the most toxic from all of them, we can target with one single molecule. And I want to tell you how that works. So that molecule drug is called antisense oligonucleotide, or ASO. I guess you already know a little bit about this. And so, but I'll tell you anyway. So these ASOs are small molecules that are very specific to a certain RNA. So if you have an RNA that you want to get rid of, you can design a molecule that's very specific. When the ASO finds the RNA that you're interested in getting rid of, there'll be a protein that comes, and it's like a molecular scissors that cuts the RNA in small pieces. So get rid of the RNA. If you don't have RNA, you're not going to have protein. So the way it works in the C9 situation is that we design the C9 ASO that is very specific to the disease RNA. When this ASO finds the disease RNA, again, there's a protein comes and cuts it and gets rid of that. If you get rid of the RNA, then the RNA, it's not going to accumulate in the cell, and it's also not going to be used to make disease protein. So with one drug, we can eliminate two possible sources of toxicity. So let me show how that works in the cell. So we have a Petri dish with neurons, and we're going to treat them with ASOs. And then we're going to look at the disease molecules. So here we have, so in this experiment, we have 3 independent Petri dishes. One we treat with a control ASO, which means it's an ASO that does not target any RNA, which is the bar here. And then we have 2 other Petri dishes. Each one of them we're going to treat with a different C9 ASO. And as you can see here, when we look at disease RNA, after treatment, there's a large decrease in the amount of this disease RNA. We also looked at disease proteins. And again, this is the same experiment. After treatment with the ASO, we saw that was a large decrease in the amount of disease proteins in our cells. So this is the way it works in human cells in a Petri dish. And now we would like to try that in human cells in a human body. But when we do that, there's many things we have to take in consideration. Today, I only want to talk about one of them, which is when to begin the treatment. So if we think about this graph, which shows disease progression over time, and the dashed line is the diagnosis, what happens is disease starts, and the progression of this is dependent on the cells that die. The thing is, we don't know when the disease starts. We know that a person has a disease when they first show their symptoms. But by the time these people showing symptoms, there's a lot of cells that died in your brain. Once the cells die, we can't bring them back. So we have tried, there was a trial that tried the C9ASO. This was a clinical trial, and it was trying on people that have ALS. And the people were treated with ASO, but after a while, it was found that the disease continued to progress, and so it was discontinued, it was stopped. We don't know exactly, there's probably many reasons why we didn't see the results that we thought we would see, but one possibility is that maybe at that time it was already too late. So again, if cells die, you can't bring them back. So maybe it was too late to do this treatment. So one thing that we are doing now is we need to find a disease marker so that it can tell us when the disease starts. Because if we do have the idea when the disease starts and before the person develops symptoms, we could potentially start this treatment much earlier, and then we have more chances of have a bigger impact in preventing the disease to continue to progress. So today, what I told you about, so this is summary, I told you about FTD-ALS, that is caused by loss of brain cells in multiple regions of the brain. The most common genetic cause is a mutation in C9orf72, and there's 3 ways that we think that mutation can lead to the cell, can cause the cells to die. And I also told you about an ASO drug that can block 2 out of these 3 sources of toxicity. And currently, we are in need of disease markers to help us guide the best time to start this treatment to help people that have these diseases. And so finally, I would like to thank all the donors and their families for providing samples and making this research possible. I would like also to thank all the sources of funding that help us during these studies. Thank you very much.
Instead of using markers of the disease, could you just use the genetics and look at people who are at very high risk because of the mutations that they have in C9?
Yes, we could. So if we know the person has a mutation, We could potentially think when we could start the treatment, but no one wants to start the treatment when you don't know if you're going to develop disease or when. So I think, you know, and the other thing is like, you know, people that have the mutation, it doesn't mean that they are going to have disease at 100% of the time. So you don't want to start the treatment and you don't know if people are going to need it. So they are at high risk. If they have mutation, they are at high risk, but it's not for sure they are going to get the disease and when. And so these treatments may not, you know, this is not like a little pill. This is kind of food infusion, and people have to take it multiple times. So you want to take it only if you really need it.
So this is very nice talk. I have two questions. So firstly is for your third consequence of the repeats, how do the disease protein do bad things to the patients?
Okay, so these are abnormal proteins. We don't know what they do. Some of them have a charge, and they tend to accumulate in the cell, and they can interfere with normal function. So they can go to the mitochondria. They can interfere with transporting the nucleocytoplasm. So many things that, you know, there's so many studies, and these cells seem to be very toxic for the cell.
My second question is like, if I want to diagnose whether I have a mutation, like what do you think is the best way to get earlier diagnosis?
I would say probably would need to go see your doctor, maybe talk to a counselor, because some of these diseases, they don't have cure. And so do you really want to know if you have it or not? So it's something you need to think about.
So please do correct me if I'm wrong, but my understanding with ALS and FTD is that most cases are sporadic, meaning there's no known genetic mutation associated with the disease. How would you go about trying to identify diagnostic markers or develop treatments for patients where the underlying genetic cause is completely unclear?
So for us, the majority of the cases are sporadic, so we have no idea what really causes disease. Our hope is that by studying some of the genetic cases, it gives us an idea of things to look for. And so if we find, I'm not saying like this particular ASO because it's very specific to the C9 mutation, but we find ways to block the disease in a mutation situation, we could probably use that as well for the sporadic. But they are in the disease, accumulation of disease proteins, TDP43, for instance. And so those could potentially be used like a diagnosis. When do these proteins start to show up in the brain? What happens at that point? I think we could use maybe some other markers, there are some things common, you know, in the sporadic, but we don't really know what causes the cells to die. So we just don't know.
Do all these cells have all the neuron cells have this issue? Because like once you create new cells, they might not have this problem for a long time, let's say 45 years.
Right. So in terms of the mutation, so all the cells will have the mutation, right? So all the cells will have the mutation. The brain cells, if we talk about neurons, these cells, they don't divide. So you are born with the number of cells you're going to have for the rest of your life, if we're talking about neurons. But all the cells in our body, we will have the mutation. What is really interesting is why only those specific cells in your brain die and not the other ones.
I have a follow-up question about the mutations. For those individuals who do have a mutation in the C9 gene, are the same mutations associated with either FTD or ALS, or do different mutations result in either FTD or ALS? Or are there other factors like maybe environmental factors that will contribute to the development of either disease?
Yeah, so that's a very interesting question. So C9 mutations can cause FTD only, ALS only, or a mix of both. And we don't know why, because as you saw from the presentation, like there's different brain cells that die. So why in some situations you have the C9 mutation causing cortical cells in your frontal temporal lobe to die, and in ALS, for instance, in a spinal cord? So we just don't know. Yeah.
Any other questions? No, if not, we'll thank Dr. Sandra. Thank you. Okay, with that, we will end the session. So I will now do the final slide. Thank you so much. All right. So with that, I would like to thank everyone here for attending. I would like to thank folks on Zoom also for attending and for asking questions. We also would like your feedback. So if you could please take a minute to scan the QR code that's on the screen. And for folks on Zoom, there will be the link added directly to the chat. If you could please take a minute to answer a few brief questions about this event, we would greatly appreciate it. We always strive to improve our events. So please let us know if you have any suggestions, and if you have any suggestions for topics for future science cafes. Also, we have a variety of social media. So if you would like to stay in the loop with our upcoming events, please follow us on social media. Again, thank you all for coming, and we hope to see you at our future events. Thank you.